Hello everyone, today we're going to be looking at the IB Chemistry SLHL topic 1.1. This comes under structure, and the specific subtopic is the introduction to the particulate nature of matter. So let's start. So how do we identify different solids? So solids, up here we have impure solids as we have pure solids. Now, what exactly is an impure solid? So this would be a mixture. Now, what is a mixture? It's when you have something combined in a non-fixed ratio. In the IB, there are two types of mixtures that we need to know about. There's a homogeneous, which is when the composition of a sample is consistent. Composition is the same throughout sample. An example would be dissolving in ACl or table salt in water, because that dissolves quite nicely. Now, the other one that we need to know is heterogeneous, which, as you can assume, is the opposite of homogeneous, so composition is not consistent throughout the sample. And a good example would be putting oil in water, because hopefully, as we all know, that makes little bubbles. Now, into our pure solids, we have elements. Now, if we look at elements, these can be atoms, but these could also be molecules, as long as they consist of the same atom. So these would be your diatomic elements, such as O2, oxygen. And our other pure solids would be our compounds. And these are chemically bonded to each other. They are in a fixed ratio, and these comprise of molecules, ions, and good examples would be H2O, because as you can see, our fixed ratio here, 2 hydrogens per oxygen in this water. As we are looking at this ratio, there's also something that, this is kind of something that if you did GCSEs, you would know what these are. Don't know what the MYP is like. Mass ratio. So how do we calculate mass ratio? Hopefully, as we know, moles is mass over mR. So let's use this water example. So if we're looking at the hydrogen in water, H2, it's mass. So we're rearranging the formula here. We have 2 moles of hydrogen. And as we know, looking at the periodic table, this has a MR or a AR of 1. 2 times 1, hopefully we know what that is. That would be 2 grams. And if we look at oxygen, that would be 1, because there's only 1 mole of oxygen here, times 16, because that is the AR of oxygen. Some very accessible maths here. And if we look at this, 2 to 16, so 2 to 16 ratio. Now let's simplify this, because we like to keep our stuff simple here. Divide both values by 2, and you'll get a 1 to 8 ratio of hydrogen to oxygen in terms of mass. Now, Oh yeah, we got room for it. So, solubility. Something else that we need to know. So what is solubility? So this is the ability of a substance to dissolve in a homogeneous solution. Now there are factors that can affect a substance's solubility. So this would be the purity of the substance, the temperature at which is at, the pressure, and other co-solvents, or just other solvents present in the solution. Now, to express solubility, we can either express it via molarity, which is the moles of the solute per liter of solution. Or we can simply just do a mass volume percentage. And how do we determine, or better yet, what determines the properties of the solution? So the type and number of particles in the solution determines its properties. But what properties? What do we mean by properties? We mean boiling point. We mean osmotic pressure, which refers to the amount of pressure, or change in pressure required to stop osmosis. And also freezing point depression. Freezing point depression. Now, if we look at freezing point depression, no, that's not ideal. Let's just look at water, for example, our favorite example to look at. Pure water, as you may have learned in GCSE, or generally, this is general knowledge actually, pure water will boil at exactly 100 degrees Celsius. But once there are impurities in the water, such as minerals, or maybe there's a bit of dirt in there, this will decrease. And as this boiling point decreases, freezing point decreases as well. Now let's give ourselves some more room to talk about the next little half of 1.1. Thermodynamics. Now, what is this? What is thermodynamics? Well, all you really need to know in chemistry is that thermodynamics is the study of energy and its transformations. Particle model of matter. Maybe something you've heard of before. When we say particle model of matter, essentially we're talking about the little circles that represent particles. You may have seen this in your GCSE before. You'll just, each thing like, ooh, particle. Now, why is the particle model of matter useful? Obviously, it gets far more complex than this. This is a very simplified version of what that could look like. So, this is important to demonstrate to us the relationship between the energy of particles and their temperature, which is good to explain to us the kinetic theory of gases. So, why is this useful? Because it also describes gas behavior at a molecular level. So this will be how particles move, collide, how energy is transferred from one particle to another, and how this determines the properties of the gas. So this could be pressure, volume, or the temperature of the gas. This also shows us the relationship between pressure, that's a very badly drawn heart, pressure, volume, and temperature in gases. So this will be, for example, Boyle's law, Charles' law, Gay-Lussac's law. You don't have to know, I believe, exactly what these are, but this is just essentially what it is, for people who are into that kind of stuff. So essentially, what does this mean? It's how the properties of a gas change as the conditions that they are exposed to change. Now, this is also the particle model of matter, important as it helps us understand electrical and electrical and magnetic properties of materials, which are determined by the behavior of charged particles, as well as the interactions between them. So this is helpful for understanding how magnets and superconductors work. Finally, this is helpful as well to understand the properties of biological molecules. That's molecules, not moles. So what does this look like? This is just, your proteins, DNA, carbohydrates. Please write that in full in your exams. Why is this helpful? You may ask, or you may not. Regardless, the IB wants you to know that this helps scientists develop new treatments or drugs. And this also allows them to develop new materials which mimic biological systems, which is very great. And that is all for 1.1 structure. Hope that was helpful. See you next time.